to the Weekly Bioanalysis, a KCAS podcast.
Hello, and welcome to the 57th episode of the Weekly Bioanalysis, the official podcast of KCAS Bioanalytical and Biomarker services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. My name is Dominic Moreno, and I've been in the bioanalytical, LBA, and biomarker space for over 28 years. I'm here with my co-host, John Perkins.
Hello, I'm John Perkins. I'm also senior scientific advisor. My focus is LCMS, and I've been in CROs for around 28 years. But I worked out earlier. I've been in LCMS for over 35 years.
John and I are senior scientific advisors at KCAS, and either or both of us are available to answer any of your questions you may have regarding this podcast or any of KCAS services. I'm located in Kansas. John is in New York. Our producer is in Missouri. So we kind of cover all of the map. We are thrilled to have you listening to the 57th episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcast, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you are joining us for the first time or you're a regular listener. Today's podcast, as always, will be a review of the latest news and resources, and then we focus on a topic of our choice before discussing any feedback we've had from you. We are constantly looking for topics, and we would be happy to discuss something that you want us to cover. Then we will wrap up with a little teaser for the next episode, letting you know what is coming in the future. So again, we're fired up to have you here, and we are looking forward to a fun episode. John, why don't you go ahead and kick us off with what the agenda is before we dive into the news and notes?
Sure, thanks, Dominic. Good to get together again. So, as you mentioned, that we'll go through some news and resources, which we touching on the usual process of COVID items, then some non-COVID news, and some investment pieces as well. And then our main portion today is the evolution of drug design and its impact on bioanalysis. It gives us a good chance to dig into our history with bioanalysis and where the pharmaceutical industry has gone or has come recently and how it's affected our day-to-day existence.
Yeah, John, very exciting topic for both of us. But with that, why don't we get started with the news and resources?
Yeah, well, actually, this one is a little bit not quite COVID, but obviously we've talked COVID to death for the last 2.5 years or so. But we've had the recent outbreak of monkeypox, which is in the news. And there's obviously cases rising globally, particularly in the UK. But there's a couple of analysts looked at it and said their take is that the monkeypox outbreak won't be the next COVID, simply because different features of the viruses. There's available vaccines that smallpox vaccines that can work against monkeypox because it's closely related, and it's just just talking through the different features of the virus. And although it's something to be possibly concerned about, it's not going to be the same level of effect on our lives as COVID has been.
Yeah, certainly not. I mean, a couple things. One, it's not nearly as transmissible. It's not airborne transmission. They believe it's sexually transmitted. And then there's a vaccine already available, John, right? There's a couple on the market. So, but I do think it's important to touch on things like monkeypox. Actually, I find it to be I think it's bad. There are cases, 3,400 worldwide, maybe 350 is the number I heard. I was kind of making the news a little bit. It's about 350 in the U.S.
Yeah, there was when I looked at this like a couple of days ago, it's about 1,000 in the UK. So you got 3,400. And this is really it's come out of Africa. So Europe is the hotter spot for this. So it's not a huge number of cases, but they are growing. I mean, it's not like it's 3,000 cases and it's capped. It is a growing number of cases.
But it's growing at a much slower rate than COVID would have.
Exactly, yeah.
And then the other thing, where I was going was, I think it's kind of this return to normalcy. Is this part of what happens when we go back and now we're like super sensitive to outbreaks? I mean, I don't know. I'd love to see a history of like outbreaks of things like monkeypox or even SARS and other things. Is this just a normal sort of outbreak and the ebb and flow of having to live together? Or is this, are we is this always been present, or is this just being covered, or is this really like we're now emerging into this viral thing? Probably somewhere in the middle, right? Probably a little bit of both.
I think with monkeypox, it has been out there, but it's not been a it's not really broken out of Africa like this. I think you hear of like the occasional cases in like Nigeria and places, but it hasn't really broken out the continent. I think that has made the difference, and then there's transmission of it. But I mean,
but are we testing more? See, I don't know if I think this is an interesting debate, right, John? Is it that 10 years ago maybe there was some cases in Africa and people thought, okay, we'll just, and then we were running around getting it and no one really reported on it, or maybe it did, like we don't, but clearly now we're like heightened, right? We're sensitive to it.
That could be the case because obviously Africa doesn't really feature.
Again, I think it's a good thing, right? I'm not saying it's not a good thing. This is progress, but what I'm trying to say is like, I don't know how serious an outbreak 3,400 is relative.
Absolutely, and I think another part of that is if you looked at the when cases were first being diagnosed in Europe, and people said, okay, we've got cases of monkeypox in Europe, but ultimately it's usually non-fatal, it's got a low transmission rate, it doesn't mutate very much, so it's not going to be all these different variants. So I know it's at least in one of the articles that said that the bulk of the virus was sequenced, and it's shown it's pretty much the same as outbreaks in 2018 and 2019. So it's not doing like COVID did, where we've gone through numerous variants.
Yeah.
So I think right in the early days it was much more level-headed assessment of what monkeypox is. It looks unpleasant. I mean, you've got all these blisters, etc., then break, etc. But ultimately, it's not a health threat to the scale of what COVID has been. But it's just something to be aware of, and I think, like you say, they haven't really figured out how infection works, but there is a sexual element to it. So I think there is going to be somewhat of moderating of behavior.
Yeah. And then, of course, drug use would have to do it, right? Any sort of IV drug use would do it as well, right?
Possibly, yeah. That again, I don't know. it's not the.
Well,
circle.
I mean, me personally,
it's not the circles I move in. So
yeah, no, and neither do I. But I mean, let's face it. Anytime you're sharing a needle, you can transmit any disease, in my opinion.
Oh, sure.
That's like, well, not cancer, right? You can't catch cancer, I guess. But it's an interesting topic, and one that I think we could probably continue to talk about, John, because it's weird how for me, I'm standing here, I'm like, wow, our minds are really thinking differently post-COVID, right? Like it's really a strange thing, a strange phenomena how we're thinking.
Yeah.
But good stuff all around, and looks like John, why don't you tell us a little? One of my favorite subjects, right? We got T-cell assessments, or.
Yes, so I think I was pandering to you a bit when I put this one on, but again, it's interesting in terms of we're still talking about how we assess the effectiveness of vaccines and things like that, and this was biotech academic leaders encourage FDA to consider T-cell assessment in COVID-19 vaccine trials. So obviously, where everyone's been talking about effectiveness of vaccines, we talk about the antibodies we have in our system, etc. But in a look, this letter is really saying, the following vaccination, you see a decline in the immune response, but you don't see a decline in protection against the disease. So they're saying T cells should really be a much bigger part of the conversation than they are. And I'll pass it on to you to say your piece.
Yeah, early on, I think I kind of emphasized the thoughts around T cell immunity, and even I still think there's something to be had in that ACE2 receptor kind of pathway there. But this is a very fascinating story in terms of, the vaccine is designed to really augment antibody production, right? And so we're still trying to figure out if it can generate protective immunity, which, by the way, is that's a big statement I just said there, protective immunity, right? Or central immunization, or creating central memory T cells. That's still kind of a holy grail of immunology. And so the vaccines themselves, they certainly, you can't get antibody production without T helper cells in particular, right? You need some aids and some help, but that helps the B cells produce antibody that turn into plasma cells. So these vaccines obviously have some T cell effects, but not like a natural immune response where you're truly going through that innate to adaptive to triggering central immunity or central memory immunity. So I mean, the hopes are the vaccines, and I think that's the next thing. Like, I'm excited. I'm like, oh, let's generate vaccines that can do that. But this letter is really going towards that. Like, let's start to offer tests that are going to make people feel a lot more comfortable about their health, John.
Yeah.
Sorry, I know I went a big full circle.
No. I appreciate it, because I wanted your input on this. I mean, they're basically saying, let's do some effective testing to pull up, to basically accumulate T cell data, so we're getting a good understanding of both the antibodies.
And you know what the problem is, right? You can do an antigen test, a swab, right? They swab you. That's a lateral flow test by ligand binding assay. That's what that is. Or you can even swab and they can do PCR. What do you need to do to understand if you've got protective T cell responses for COVID? John, you know where I'm going here. What platform you got to use?
I haven't got a clue. I mean,
it's flow cytometry and ELI spots. We're right back into those hard hitting platforms, right?
Yeah,
those are the platform. There's only two recognized gold standards for measuring T cell responses, functional T cell responses. There are other platforms, but the gold standards are flow cytometry and ELI spot. And that's why we've moved in our cell and gene therapy that way, but it also has really a lot of benefits for vaccines and infectious disease as well. So those are the platforms. And really, this is a cost thing, in my opinion. That's why it's just an expensive thing to run.
Sure. Yeah, absolutely. Yeah.
But it's John, I would really love to know. Imagine if when you go in and your blood test, oh, hey, you know what? The likelihood of you getting COVID is almost negative because you've got circulating T cells against COVID, right?
Sure. So is it expensive in terms of being unautomatable or
well, it's a couple things. You need it's just the whole process. You need a sophisticated flow cytometry panel. So first, someone's got to create the in vitro diagnostic flow panel or ELI spot panel, and that in and of itself is a much more longer road to haul and harder to approve than, say, a lateral flow swab test that you stick up your nose and you put on a, you know what I mean? Like the ones we take for the COVID antigen.
Yep.
That process becomes harder. You need then the whole entire, the reagents for it are 10, 15-fold higher. You got to buy a cocktail of antibodies that are labeled. You need a sophisticated piece of instrumentation, and then you need a really trained technician, and then the reportables itself. So even running it, doing the reportable piece takes an even more skilled individual, right? So you got a skilled person running the test, and then you need even a more, and I'm not talking about doctors. These are like people such as myself, or even like lab director, senior scientist, that type of level to report that data. Principal investigator. So you got a whole series of things where, these antigen tests, they come in the mail. I'm doing them in my kitchen. We're not doing flow cytometry in the kitchen.
Sure. Yeah. Did they also have Jeff talked last time about, so many of the samples, you can't freeze them. Would that also be the case here where you're talking?
Well, it's even more expensive to freeze down a PBMC. Now you're adding costs. So the answer is but you're making it more expensive to batch them. So a whole blood test is a fraction of the cost. Approximately probably half, getting a PBMC and putting it in liquid nitrogen has cost to it. But good question. But a whole blood is the way to go. It's just, I think there's got to be some hematology analyzers that can solve this for us. If I'm a hematology company making, hematology analyzers, I'm looking for a quick and dirty, because a lot of them have the ability to measure CD4 cells for HIV, right? So a lot of them kind of have a flow cytometry component to them. They can tell you a 3-part differential, and then they can give you a CD8 and a CD4 count to help with HIV. Well, if they can do that, there's probably a way to do a COVID T cell test, but we're lacking, right? That, like I mentioned, the diagnostic isn't there yet, so therefore, by default, the hematology and flow companies can't really do much. But they're out there. They're getting there.
Sure. Absolutely. Okay.
Yeah, I'm fired up.
Shall we move on to the non-COVID?
I guess so. Yeah.
Okay.
Tell us a little bit about.
That was really helpful. Thanks. I'm glad I mulled over whether to include that or not, and I'm glad I did, because that was. No, helpful.
I thought that was a great one. The academic leaders encouraging the FDA to do because that's, John, we all want the same thing. There's a lot of disputes about COVID vaccine mandates and all this stuff, but what we really want to know is, are we okay? Doesn't matter what your political affiliation is. And that's what this did. This moved towards a step towards, hey, let's find a better technology so that, if you're an 80-year-old, you're going to feel a lot better, right? Like it's not necessarily for me as somebody who's still relatively young, but I couldn't imagine if I were like, my mom, for example, my mom will not leave the state of Massachusetts, right? This would be helpful
Yeah.
for someone like her.
No, I agree. You definitely, we want to be figuring out how can we tell people that they are protected and able to do things, etc.
Yeah, instead of hearing like my nephew who's not vaccinated saying, "Oh, I've caught COVID twice, so I'm protected." Are you? I mean, I think maybe, but I don't know. When it comes to my health, I don't want maybes.
No, exactly. No, absolutely not. No. Yeah. Good luck with that, boy.
Yeah. Yeah. Well, he's, well, anyhow, let's move on.
Yeah, let's move on. This is just a little snippet. The headline of this is "Talk to your doctor. Pharma TV ads are still spurring patients to action, study finds." And if you're watching cable, particularly, always seems to crop up. But there's a lot of ads for various medications, and as a sort of someone who's involved, I find them fascinating because I always want to know what the drug is behind it, whether I've worked on it, and who the manufacturer is. So that's really where I come from, and because obviously we read a lot elsewhere, generally there were these things out there, so I'm not looking to those ads for information. But it was interesting. It was a survey that talked to, I think, 3,900 people, 2,900 US adults, to get their perception of these pharmaceutical ads on TV. And there's actually a reasonable proportion, around 27%, actually talk to their doctors about potential treatment based on these TV ads. So although they might, you sort of wonder, why is a pharmaceutical company advertising? There is some effectiveness to them. And I just thought it was interesting to have this, pointed out in a survey that actually there's some benefit to it.
Yeah, John, I'm like you. When I see an ad, if I don't know we don't always know the commercial name of the drug, so I do like to Google it and be like, oh man, I have worked on this, or oh my God, we've worked on it at KCAS. That is really what I'm focused on. But it's interesting. I think it's really interesting to hear that 1 in 4, about 27%, will talk to their doctor. That's a good thing, John, right? I really, absolutely. Most of the people in my life, when they tell me something about a doctor's visit, I'm like, well, did you ask them any questions? And they don't. And I'm like, well, I actually kind of take the opposite approach. I look at my doctor and say, oh, now when I walk in, he's kind of like, he twitches a little. He's like, okay. Mr. Worry, Dr. Worry, I don't want to hear too much from you about vaccines today. I'm short of time. No, but I kind of joke, but I think that's fantastic that it's spurring people. And then it actually said, what was it? Almost 60% had a favorable response to the ads, right? Yeah. So good for the pharma industry here. They still get a bad rap. I did read in one thing they kind of plummeted. Remember, they kind of
they have. Yeah,
but this is good to hear. I mean, John, all these ads, sometimes you are like, why are we spending money on this? But if it's caused 1 in 4 people to go in and ask better questions to their doctor, then I say let's pay for it, right? That seems like a good spend.
Absolutely. I mean, we're in a sort of fortunate position that we're, like I say, we're aware of what's coming through and potential. You're looking at drug approvals and things that are coming through. For us, my wife suffers from migraines, and so she's gone through a number of she's done sumatriptan, fevertriptan. She's currently doing Botox to help with migraines, but it is definitely an area that I'm always watching for what's coming through in terms of potential medications that may help in the long term, because I know how disruptive migraines are to her day-to-day life and by extension, the family's day-to-day life. So I'm like you say, I'm all in favor of this. That if it really is that larger proportion of people react to these ads, I want to see them on TV because it can only be a good thing.
And it's funny, my wife suffers from migraines too, and I was just looking for I think I was looking for an aspirin or something, and found a whole bunch of what you just described. So she's got a litany of it as well in terms of migraines. And you're right, where it'll ruin a day. Like one time we were on vacation, she was very fortunate. She had some a sumatriptan injectable she had. And she needed an hour and then she was fine. Otherwise, we would have lost that whole day, John. And look, I want my wife to be healthy. It doesn't matter. We couldn't have gone to Disneyland or wherever we might have been, but that's what it's about. It affects everybody. And that's another. We're going to move on because we're kind of going long, John, and we got a couple other topics. But
I,
migraine medications is another thing that's come a long, long way, John.
oh, absolutely.
Great advances there.
Yeah.
But let's
Yes.
move on to what do we got? Small rectal cancer drug.
Oh, this is amazing.
Oh yeah, this is a good one. Even my crazy brother liked this one.
Small rectal cancer drug trial sees tumors disappear in 100% of patients. Now, this got into like the national media because of obviously this is incredible. It is a small drug trial that has to be emphasised. It was basically 14 patients who had. Actually, it's a specific type of rectal cancer, which I know is on here, and I can't find it quite yet. But anyway, 14 patients. They all had the specific type of rectal cancer, and as they were monitored and by the company, all of those 14 patients had the symptoms disappear, which is, I mean, absolutely incredible. It was an antibody drug called dostarlimab. And it was developed by a company called Tesaro, who have since been bought by GSK. The actual study was done at Sloan Kettering Cancer Center in New York, and it's basically all 14 patients were diagnosed with early stage rectal cancer, and the 14 that they cited had completed the study by the time of publication, which is the New England Journal of Medicine, and it's seen as a treatment that could actually point the way to treatment for other cancers in the future. So I'll pass that back to you while I figure out what the actual. It's great anyway.
Yeah, John, I couldn't find it fast enough for you on what the actual colorectal type was, but the N number is somewhat low, but it 14 is still, it is what it is. 100% is still fantastic.
100%. Yeah.
It doesn't. This is clearly they use the word groundbreaking, and unprecedented is so overused these days. But this is truly some unprecedented, groundbreaking stuff. I mean, trials simply don't go 100% like this. There's something clearly is connecting in terms of this subset of colorectal cancer patients and this PD-L1, right? That's what this is.
I actually have it, so I'll just, because I'm going to quote the article directly. The study does have caveats. Sample size of patients, while diverse in age, range, and ethnicity, was small, and even the earliest patients in the trial still have several more years of observation to ensure that the tumours haven't re-emerged or metastasized elsewhere in the body. The results also only pertain to those who carry a specific abnormality to the rectal cancer known as mismatch repair deficiency, which impedes the body's function to normalize or repair abnormalities when cells divide and instead results in mutations. And that deficiency occurs in roughly 5 to 10% of all rectal cancer patients. But it tends to resist chemotherapy. I think that actually is quite a key piece. This is, although it's only a small percentage, it's tough to treat cancer, but this antibody is at least in this one trial was 100% effective against it. So yeah, it's a big deal.
Yeah, I think it leads nicely into some of the topic we're going to talk about, and in terms of drug design and just in general. I can't tell from this article if these patients had already gone through the standard regimen of radiation, chemo, and surgery, and then got the antibody. But what we're starting to find out is like they're, I don't want to steal too much thunder, but I'll talk about that in the main section.
I think reading it that they hadn't gone through any radiation and chemotherapy. They were actually planning to go to radiation and chemotherapy after this. So they were on the trial, and then it was next. And so they cited one patient, which I haven't copied into here, but she was going. I think she was based in Boston, was going to move down to New York, and she cancelled the move to New York because she didn't need either the radiation or the surgery that was going to be scheduled normally to deal with this. So yeah, it was it made that difference to her life,
Yeah, John, it's even though it's 5 to 10% of colorectal cancer, I think that's the subset they're talking about here. It's just awesome. This is exactly why we got into medicine. It's in the 1980s, the monoclonal antibodies were on the cover of Time. Well, here we are in 2022, finally with really. There's been lots of lives saved with monoclonal antibodies, but this particular one is an indication of how refinement in the technology, advancements in the technology, making it bigger, badder, and safer. That's what this is all about. Because it's
Sure,
like a
absolutely.
next wave of PD-L1, PD-1 antibodies. So great. I mean, I'm like, this gets me so jacked up to think about bispecific antibodies, ADCs. I mean, we're scratching the surface with these, but this is a
Sure.
really groundbreaking study here. It's great.
And just one quick summary. There's a guy called David Ryan, who's director of clinical oncology at Massachusetts General Hospital, and I'll cite Andreas Serchik, who was the lead on this study. They both said that this could have, although this is rectal cancer with mismatch repair deficiency, there are other cancers that use that mechanism, like or other tumor types like pancreas, stomach, or bladder. So it may be a similar approach to be used for those. So that's where the potential lies.
Definitely, John. Anything,
it's not just 10% of rectal cancer patients. There's a pathway where you can go if this in this direction that could really help.
I'll take it a step further, John, because I studied cervical cancer. It's a mucosal immune system cancer. All the ones you're talking about, that's from the mouth to the anus. That's its own little immunological sort of prevade, sort of it's its own little system within your body, right? And so you're absolutely right. Stomach, bladder, pancreas. I'm going to even say head and neck tumors, which great advancements in them. But it's got to be anything from the mouth to your butt is going to be impacted by this. I said butt that time instead of anus. Is that okay to say? No.
Oh, that's fine. Yeah.
I heard a little giggle in there, which made me laugh a little. But no, all kidding aside, you're nailing it, John. This is like, and if you can unlock 5 to 10% of a subset, then the ability to unlock 100%, or 100% is a little unrealistic. But there'll be a day, hopefully before I pass, that 9 out of 10 cancers are treatable in like mucosal immune system. That's within that mucosal. I call it mucosal immune system, but from your mouth to your anus there. All of them are having great, I'll call them, better to have, advancements daily, John. And again, in our lifetime, I think these types of approaches in conjunction with, maybe put a small molecule payload on it, or even in conjunction with some sort of gene therapy. Imagine a CAR T cell therapy in these patients along with this monoclonal. Sky's the limit for curing people. It's fantastic.
Sure. Yeah.
Well, I think we got to move on, John, right?
We do. And these two articles really both flow together. And it's just touching on attention turning to ALS. And first of all, there's discovery of ALS and dementia mechanism points to new treatment strategy. And the second part is the FDA unveils 5-year action plan for neurodegenerative drugs. As our population gets older, we're going to see more and more of, and I've seen our relatives affected by, obviously Alzheimer's, dementia, and other such diseases. And ALS is one that hits people earlier in life, but it's still something to be concerned about. I mean, ALS was in the news recently when we talked about the Biogen drug for Alzheimer's, where I think it's Excella have a drug that they think is effective, but there's no biomarker to actually indicate what it's doing. So it sort of stumbled a bit in the approval process. And so that's part of where the FDA strategy is coming from. But anyway, to go back to the discovery of the dementia mechanism, almost all cases of ALS are linked to dysfunction in the protein TDP43, and there's 2 teams have linked that problem with one of the strongest genetic risk factors for the disease, revealing a possible biomarker, a new therapeutic target. So if you lack TDP43, it basically could quash the expression of a neuronal protein called UNC13A. Things get worse when UNC13A genetic variants are present, increasing the risk to more severe ALS and FTD. So by identifying this protein and the improper expression, it may give another pathway to actually try and address this disease. And like say, then gives a biomarker where you can actually look at the effect of your drugs and are you helping things go the way they should.
As I sit here hearing about this, it makes me think about even within my lifetime, it's been no drugs really. When I first started in the 90s, wasn't really talking about neurodegenerative, maybe a little bit. Flash forward 10, 20 years, You've got all sorts of investments in drugs that target like tau, right? You've heard tau or phospho-tau or
Yep.
even things like A beta. And then what they found out is they make these networks, and they're still trying to unlock that. It's such a hard thing because you can't take a piece of someone's brain. If you noticed, a lot of the research is done post-mortem, and that even that it's great to look at, but is that really, there's obviously any sort of cadaver will have lower levels of a given drug or whatever, biomarker or whatever sort of protein you're looking at. I don't know if you knew that, right? That makes sense. So they're probably under, even when you study these things, it's still not a fresh sample. So what am I driving at? Despite what, the Biogen aduvumab sort of kind of, I think it's kind of been taken back off the market, John. I don't know where it stands, but
Yeah. I wasn't dwelling on that too long.
Through all that, I think it's still a new. I think it's still something. It's still new, right? We think. Sure. You heard me say monoclonals have taken like 40 years to get to where we're at. We're probably 20 years away from making real breakthroughs in the neurodegenerative space. Hopefully we'll do it sooner, right? But this is these types of articles and these types of findings around new biomarkers. They got some great names like UNC13. I wonder if that's, really the UNC I'm thinking of, but these are TDP43, all of that. So it's like the next wave of biomarkers. So I'll end with, this is promising. And then I love that you got these African nations kind of going into this space with the FDA as well. So it's a global health movement. Yeah. And then I said I was going to end, but John, I know all of us are certainly, we're all living longer, right? The heart's going longer. We're all eating and exercising and body by chemistry. We're going to live longer and longer. But the brain, man, I'm doing as many Tetris puzzles and crossword puzzles as possible.
Yeah. So yeah, I mean, it's I agree with that, and the reason why I wanted to include this, because I still feel with everything I read, we still don't really. There's so much work being done in this area, but it's still really trying to figure out what is the root cause to these diseases. We're not there yet. Like I say, I agree with you that we're several years off the medication that's going to fix this. I mean, I picked this one up, but there was also another article which I didn't include because obviously time, etc., that was talking about in terms of, for Alzheimer's, we talk about plaque, but maybe we're not looking at that early enough. And there's a research group have some thoughts on that. And maybe I'll
Well, no, but that, by the time that plaques form, it's progressive.
Yeah, maybe I'll pull that article.
And what they're trying to do, the other part of that is each plaque, not only they're trying to define what's in those plaques. So say mine is a combination of, non-phosphorylated tau, but a percentage. I mean, it's crazy some of the things I've read. And then the amount of P42 relative to, the amount of A beta 42 versus A beta 38, meaning you could have high levels of 42 and low levels of 38, and that's bad. But if you have low levels of 42 and high levels of 38, that could be good. Think about that, John. That gets into a whole gray area around how sensitive are these methods. And then we're taking plasma samples or maybe even CSF samples. And that's still a lot of questions around that blood-brain barrier. It's beautiful. The blood-brain barrier is a beautiful thing. We've made major advancements, but it's there for a reason, John. And it's hard to get to the brain, as it should be.
Sure. So anyway, yeah. So that's the potential discovery, which I think it leads into the FDA piece.
All right.
And basically, there is an urgent need for research in this. So the FDA is hoping a new 5-year action plan, which is a specific detailed blueprint for ALS, can help accelerate new medicines. So they've established a rare neurodegenerative disease task force. A public-private partnership focused on speeding up the creation of disease-specific science strategies and leveraging ongoing FDA regulatory science events to boost scientific advances. I mean, it's got to be a good thing to. I mean, obviously, we look at the pharmaceutical industry. There's so much focus on oncology, etc. The neurodegenerative space is a bit of a graveyard for pharma companies because we struggled to get effective medications to market. So I think it's good that they're talking. Let's have a task force that really focuses on this and see what comes of it. And I mean,
There's a lot of players here too. Pfizer and the Bill and Melinda Gates Foundation's heavily involved.
no, that's the next article.
Oh,
That's
I was
your Emergence thing.
No, sorry, John. I did get ahead of us there. Where do we want to go from here, John? We're getting a little long. Why don't we wrap up some news?
Let's wrap up with this, because I think this it touches on what we were talking about earlier, and the perception of pharma companies that basically spiked to a high during COVID, but is then settled back down to largely unfavourable perception in the public. But things like this, I think this is a good move by a company. So the headline is all about access. Pfizer moves to a non-profit model for drug sales in 45 lower income countries. So basically, it's part of an initiative to lower to give access to cheaper drugs worldwide. Pfizer said in late May it will provide all current and future patent-protected medicines and vaccines available in the US or EU on a not-for-profit basis to about 1.2 billion people in 45 lower-income countries. I keep wanting to say companies. So far, Rwanda, Ghana, Malawi, Senegal, and Uganda are the first 5 countries to sign on to this accord. This is great. Yeah. I mean, it's like we talked consistently through the COVID piece about the disparity in vaccine access between us in the US and Europe and the rest of the world, and particularly Africa has had very limited access. Well, you've got a pharmaceutical company saying, okay, we obviously want to be profitable, but we're going to cut break to these countries because they're poorer, and get access to those medicines. That is a good thing, and I'm totally in favour.
Yeah, John. I jumped that a little premature, but there are 23 medicines Pfizer's providing. I mean, this is awesome, and the Bill and Melinda Gates Foundation's helping out. This is exactly what we want, John. These countries, they're things that we take for granted, like they highlight the Group B Staphylococcus vaccine for pregnant women. That causes stillborn. This is readily available. This is something we don't have happen in the U.S. or most industrialized nations, and this is absolutely needs to be disseminated throughout Africa and not for profit. So good stuff here. I think this is some of the.
So I hope this is one of those things that if Pfizer takes lead, others follow, and like I say, it's to the benefit of everyone to have a healthy world.
Yeah, I think that also unlocks some of the. It should lead to, even just as the world gets healthier, we can start focusing more on other things that should be kind of, I call them ablated, right? Some of these diseases should be ablated. They should be gone. They should be literally extinct. Yeah. And instead they're battling those, and that's just it's taking the eye off of, other diseases, right? More.
I mean, for instance, another article that didn't make it today was, there's a malaria vaccine, a new malaria vaccine coming through that is looking to be much more effective than the ones that are on the market, and could be a real game changer. But those are the kind of diseases that we would like to be focused on, simply because of the number of deaths associated with it in sub-Saharan Africa and other places. So
well, I mean, I think for all of us who want to travel there, these are the things that, not that it prevents you from going, but you got to take a battery of shots before you go, right?
Oh, sure.
All sorts of things. So
and they're not cheap, believe me.
And then and then there's always, it doesn't make you feel good when you get pumped full of stuff. So hopefully it's okay though. John, what a great news and resources. We're going to turn our attention to the main portion now, which is very excited, right? We're going to talk about the evolution of drug design and its impact on bioanalysis. And John, so if we think about the evolution of drug design, and maybe we go back to the 80s, and as we've talked previously, small molecules dominated the day, so I think that's where we got to start, right? Small molecules.
If I can start that, I was just like, because I always like to have notes I can jump off when I'm on this, and I found this abstract from a paper in 1987 that said, although screening of natural products remains this is 87, so this is 35 years ago. Although screening of natural products remains the major method of discovering new anti-cancer drugs, newer techniques of rational drug design, computer-aided drug design, and combinatorial synthesis promise to broaden the scope of compounds available for screening. Recent changes in FDA FDA rules allow for the accelerated approval of drugs for treating cancer and other life-threatening illnesses, although the three-phase process of clinical trials remains largely unchanged. So like I said, 35 years ago, this is purely thinking small molecule, but it's also, it's thinking like, I remember like visiting, GSK in the 90s and seeing their lab with doing the combinatorial synthesis where they're cranking out, hundreds and thousands of drugs a day. But this is pre that. It's sort of talking. We'll go to natural products, and that's our first source. And then how do we adapt those? And as I was reading that, I was thinking, okay, 1987. That was just as I was in my PhD, and my PhD was on chromatography combined with mass spectrometry, which is what we rely on day in, day out for small molecule. But it was at the time, and it was at that time, there was no routine interface between liquid chromatography and mass spectrometry. So your PK or your bioanalysis was either done by LCUV or maybe fluorescence, or GCMS, because that was the GC was the easy technique to combine with mass spectrometry. We were still in a research phase in LCMS, and it wasn't until around 1990 with the commercial launch of ion spray and other electrospray sources, and to some extent APCI, because SCIEX had both of them, that then you got that ability to actually there was the ability to have routine interfacing between LC and mass spec that then meant that you could do overnight runs, and you could do these long series of injections and measure drug samples. So that was a huge turning point.
Yeah, John. So that's a good segue into this whole discussion. So it's small molecules, they're natural compounds. You've got these tools about you, and they start to evolve, right? You've got what are basically, you kind of got to put the front end, maybe you got gas, you got the GC mass spec, and then you start making synthetic versions. And then what happens? Right around there, that's when, the first time I worked on a biologic was 1994. So that's a few years later. Yeah. But in 1994, we were taking what was called OKT3, and that's it's a murine monoclonal antibody against the CD3 receptor. And they had been using it in transplantation. It still is used. So if you dose someone with this, it actually shuts down the immune system. When you trigger the CD3 receptor and you overstimulate it, it actually shuts off the cells. Because if it doesn't, that's like a safety valve. If you don't shut off the cells, you're going to die of septic shock, right? If your cells just turn on, if that didn't have this, like it's called activated induced cell death, it has a name, right? So if you treat these people with that antibody, and that allowed for transplantation. That's where I'm going. So the first monoclonal antibodies were used in transplantation to allow for immune suppression after someone got a new organ. If there was some sort of reaction, they dosed them with OKT3, and they still use it. Cyclosporin's another one. So, but we were using it in a cell and gene therapy sense. But I was way ahead of the curve at that time. We were taking cells and using small amounts of OKT3, collecting their autologous supernatants, freezing that, and then co-culturing their autologous supernatants back with their cells. Now we do something slightly different. We re-engineer the cell. Before, we were trying to use cytokines and chemokines naturally produced to augment the cell to go after the tumor. Now in a CAR T cell setting, they rearranged the T cell receptor in culture. So that was a predecessor of it eventually. But the monoclonal antibodies start to really break through. And then when we talk about bioanalysis of it, flow cytometers are around, John. And before I was on a flow cytometer, but that was a 4-color flow cytometer that took up half the room. It had to be dark, had to be cool. Well, now that footprint of that thing is, probably 1/8 the size of it, and can do 60 colors now. But I'm going to take a breath, so I hope that leads in. I love this, John. We got small molecules; they're ramping up. That's really where the science is. And then right in the background, the monoclonals are hitting, but there's still a lot to be done. We're in the Stone Ages flow cytometry-wise. We've got ligand binding assays, but the commercially available reagents are nowhere near where they're at. They're starting to get into making hollow fiber reactors. They're starting to make antibodies. That's back in the 90s as well. So they're starting to be able to produce them. Obviously, now you can produce antibodies much faster. But John, I'm going to let you take it back from me before I moved past the 90s. We're still kind of in the 90s.
So yeah, I mean, well, actually, I think if you want to touch on the change in approvals of drugs, is that a
Yeah.
good time to talk about it? Or I mean,
So John, you thank you for that. I put together a chart for a webinar I did, and in 1990 there were 25 total approvals, and at the time they're called new molecular entities. That's small molecules, NMEs, and then there are biological BLAs, biological licensing applications. So in 90 there are 25 NMEs, 0 BLAs, and 0 cell and gene therapies. In 2000, that's when we see our first shift. There are 31 approvals, 27 of them small molecule, 4 of them large molecule. So an 87% of the approvals are small molecule. 2010, 29 approvals, 20 small molecule, 9 biologics, 69% still small molecule. 2020, 63 approvals. There's the first jump, John. We'll talk about that.
Yeah.
That's a huge increase. 40 small molecules approved, 23 large molecules approved. That's still a 63%. And that's where we're at, right? About 6 and 10 are still small molecules. And then I break it down 2020, 21, and 22. 21 had 59 approvals, 38 small molecule, 21 large molecule. That's a 64%. This is where we see the cell and gene therapies pop up. 7 approvals in 2021. Prior to that, there had been 9 total. So there's 22 total cell and gene therapies. 7 of them were approved in 21, 5 in 2022. But the total number of approvals in 2022 as of May were 18, 10 small molecules, 8 large molecules, and that's 56%. So we're still hovering in that 6 and 10 are still small, but we see there's a couple things, right? And I'll let you get it. The sheer number of them has skyrocketed in the last 10 years, almost doubled. And then that cell and gene therapy component has gone from 0 to like we're on pace to do about 10 this year, but the projections are 20 to 25 cell and gene therapies by 2025ish. So I'll let you take it back. But it was such a thrill to spend a couple hours going over the FDA.gov stuff to put this together. It was fun.
It is really interesting, but I'll wind it back a bit to the small molecule, because really, and the effect on bioanalysis. I mean, I think for a long time, throughout the 90s and early 2000s, we really were just doing small molecules by LCMS, but we were learning the nuances of LCMS and dealing with drugs out of plasma. In the early days, in the early 90s, it was like, well, it's LCMS. You've got this drug out of just say plasma as an example, you'll just blast it through, and we can do run times of 1 to 2 minutes. We can crank through N 100 samples a day, and this is great. But what I was ignoring was actually the chromatography step. We were ignoring the effect of other things in the matrix and how they might affect the drug of it, and the response of the drug of interest. We weren't really looking for metabolites. That came out. That emerged more with time, particularly as in terms of drug design, there was concern about would this drug form toxic metabolites, because that was then likely to impact its development and then kill it in the actual development and not get to market. So I think for a lot of the time, and also in parallel with that, there was also the constant industry discussion about how you validated, acceptance criteria in validation, what experiments you should be doing in validation. There was several Crystal City meetings to discuss that, and so small molecule matured in that sort of time. But what we've found more recently, and we'll touch on this, that then we've gone into a phase where the drugs that are being developed are becoming more complicated, and the actual LCMS piece is touching on fractions of these larger moieties, and I don't want to say any more than that, but that's where LCMS has become much more complicated again. So we went through this routine phase, really figuring out what was going on with LCMS, figuring out what you had to do in terms of extraction, how important it was avoiding the matrix, figuring out things like the difficulty of working with urine. You think about it. You think of it as you look at it compared to plasma, you think it's a much simpler matrix. There's less in there, but actually it's more of a nightmare to deal with because you've got things like non-specific binding that you suddenly you're losing your drug of interest. You're losing it to tubes and other things in processing, and it really screws up your data.
Well,
but we all learned all that on the fly. I mean, this is we're in a much better position now where everyone's, I know all about non-specific binding, but people had to learn through that. And then we touched on biomarkers, which I'm going to back off, but that's another important piece, because that, again, there was a learning process as to how you dealt with endogenous components, because that was not, you have to approach it slightly differently from your standard drug in matrix. And I'll pass it back to you.
Yeah, John, you said something that really struck me as, urine's an easy matrix. It's like, not to people who know bioanalysis, not at all. And it's not just the tube issue is huge, sticking, but then it's the volume, John. It's so big, right? Sometimes the volume of it, and then also things like the osmolarity and so salt and pH are really high. Those are nightmares. Maybe not so much on the small molecule, but that affects ligand binding assays. Those types of things really are things we have to kind of almost get it out of that environment. So nice summary there. On the large molecule side, bioanalytically, we're way behind you at this stage, right? You got like you talked about Crystal City. Biologics are barely mentioned at that time.
Yep.
And we're still in the, how do we manufacture them? So we're almost 10 to 15 years behind, and that's sort of what we see in terms of approvals, even, right? So we're in the we're tailing end and then when it comes to the actual bioanalysis, there is still accuracy and precision. It's starting to evolve, but that doesn't really kick off until more 2005 range is when we have a hallmark paper that comes out around biologics. So, John, I feel like if we're okay, I think we've kind of touched on the 90s. There's platforms emerging, the mesoscale discovery system, electrochemoluminescence. It's not, so there's a company called BioVeris that was a competitor of MSD. That's all the ECL. So that you've got ligand binding assays in the ELISAs are 60s, but they're getting refined in the 70s and 80s. We're making better commercially available reagents. You got antibodies that are labeled with HRP, and there's antibodies with AP, alkaline phosphatases. So you've got these beautiful, those are, from even within my lifetime, an ELISA has gone from, did this kind of work to, it's, you buy one off a kit, it's almost guaranteed to work. So that's been a major refinement. And bioanalytically, we've gone from, we've got sophistication and instrumentation around Spec readers, right? Like, so if you remember in your old school, you'd put a Spec reader in a tube, right, John? You remember doing that? Yeah, those days are way behind. We're not putting things in tubes. We're using 96 well plates. That was a huge advancement in the 90s, even plate washers, simple things. You used to have to wash them by hand and flick them, and now they got programmable things that do great jobs. Advancements in buffers, advancements in like just general camaraderie in the field to take those. First ELISAs might have taken a couple days to do. We can get one done in half a day now. Just some general other platforms. Luminex pops up in the 90s. So Luminex, mesoscale, some of the standard stuff, but those platforms are still around in the 90s, but they're not routinely used. They're still in that academic research. It's not until right around, again, about 2000, between 2000 and 2005, if we want to jump into that, John, that's when the biologic space kicks in bioanalytically.
Yep. Yeah. So if I just jump back to where we are with LCMS, like I say, there was a lot of focus on the sample, etc. But in terms of the mass spectrometers, if you look at first of all, I'll start with the chromatography. There's obviously a lot of focus on the column manufacturers and improving their chemistries, and so that improves through the years. And then in mass specs, the big gains for us have been sensitivity, where we can go to lower and lower quantitation and detection limits. and we can do it based on much smaller volumes of matrix, which is again a big deal because it helps drive the ethical treatment of animals. You're sampling patients to lesser amounts. This is all really important, and so that's where our gains are. And then another piece of it is the emergence of high resolution mass spec. It's been out there, but combining that with LC, and that's obviously we're not there yet at KCAS, but there's a number of other companies are. It's growing in acceptance because that high resolution improves your chance of getting away from other interferences in the sample and being able to detect species in samples and then thereby quantitate them and give better readouts on studies. So that's where mass spec's changed. It's really, you're right, it's that continual improvement of technology that's made our lives much easier.
Yeah, so I'm going to pick it up from here. On the large molecule side, similarly, on the small molecule, it seems like the platform, the instrument, the extraction process is important, but those instruments are advancing. On the ligand binding assay side, it's funny, the instruments, they've made advancements, but the advancements have really been in one that struck me. I'm like, one of the biggest advancements was being able to break down a monoclonal antibody, right? So monoclonal antibodies have basically, they have a heavy chain and a light chain, right? And they got two of each of those, and they form this sort of arm. But within that arm, there's a bunch of regions that they learned how to digest them with papain and pepsid. Some of the biggest advancements came from being able to take this monoclonal antibody and develop what's called an FC piece. You can knock off the tail piece of it. That's got immunological activity to it. So you can create this FAB. And then that FAB fragment has sort of two arms to it, and you can digest it further into a single arm. So you kind of got this intact monoclonal antibody that you can break easily into this dual binding region or a single binding region, and you still got this FC piece. Well, why am I explaining this to you? That actually has helped us tremendously bioanalytically, because when we go to make an antibody against an antibody, so an anti-id they call that, you knock off that FC region and it improves the immunogenic chances of creating an anti-id. It also helps with how we're building ligand binding assays. We often say if there's too much FCs, those are those little pieces I kind of mentioned, they wreak havoc within a ligand binding assay. So those are some advancements that have been used to help improve ligand binding assays in general. So I think it kind of struck me even as I was sitting here, the basic ELISA reader that we're using is really not that different from what they were using in the 60s in terms of it absorbs and emits at certain lightings. Yeah, sure, it's become automated, where your technology, I think, has exploded.
Yeah,
I'm going to finish up. We do have electrochemoluminescence and all that, but for us, really where it comes from is those, I keep harping on that, commercially available. They learned to make antibodies that don't cross-react against non-human primates. So boom, now you can detect a humanized antibody. So that's another advancement. It's taken monoclonal antibodies and making them almost 99% human, right? And all of that creates this wave that we're feeling in right around 2010 that's, I think, even still continuing to increase in 2022. All right.
Yeah, I think that's where I always struggle with LBA techniques is, I come from an LCMS point of view, where if you're being asked to solve a problem, there are so many bits you can tweak in the process, but it's all in the analytical process. Whereas your analytical process is you either get it or you don't, because your actual all the important pieces upstream of the analysis.
John, you heard me talk about it. Critical reagents, that building block, that capture antibody. We can't, if that antibody's not good, we're in trouble, right? We can try to put the antigen down, but that becomes very challenging as well, because it doesn't have the reproducibility, the accuracy and precision sometimes suffers in that setting. So it's good to be talking about this. John, we're sitting here, we're crawling up on the hour. Any thoughts on, maybe we could split this? You want to?
Yeah. So I can talk about some of it is quick. Maybe we do, but certainly one thing that we've seen recently is that drug design to improve PK characteristics.
Yeah, let's talk about that, and then we're going to cut it off, and this is going to become a two-part series.
Okay, that's absolutely fine, because a really good example of this are the anti-diabetes medications, where the first in the series was exenatide, which is based on exendin 4 from Gila monster saliva, and so that, I think it was Amylin with the company who came up with that years ago. I think the peptides of a few thousand molecular weight, but there's since been more developed where to what you're doing, you're adding pieces to the to the basic amino acid chain to sort of try and improve its half-life and retention in the body. And this is just an example. There's other medications where this happens. But what for us the challenge is as bioanalysts is when you're given the basic exenatide that's got very little alteration, it behaves like a peptide should. Once you add these lipid pieces, all of a sudden the chemical nature of the molecule, you've got the big lipid piece, you've got a peptide piece, and they're working against each other. So now you've got it drastically it affects the molecule, how it behaves in extraction, chromatography. You're starting to see carryover because it's sticking where it wouldn't normally, and that's where our issues now come. It's these designed molecules they're being designed with a particular biological process in mind, but the bioanalysis is having to pick up and run with it, knowing that our lives have just been made really, really difficult because of the alteration of the molecule compared to what our expectation would be. So that's it's a fascinating piece of where the drug industry is going.
Well, John, just because yeah, John, they made things a little harder on you, but better for the patient, right?
Oh, absolutely. Yeah. I'm not complaining. I love a challenge, and I love a challenge for our ops team. I don't have to worry about it, but it's certainly interesting when you see, because it's really important for us when we're talking to a new prospect, show us the structure of your molecule, and you see some of these things and think, oh my God.
You're like, yeah, John, I do get to cross over in your world, and I know enough to be dangerous, but sometimes I do look at something like, what is active here? Like, holy crap, this looks huge, right? Like, but then, in our world, I'll pick it up. We do pegylations, so same thing.
Sure, absolutely.
I know you pegylate on your side, but pegylations, there's been all sorts of advancements, and that did come about in the early 2000s. So what do I mean by that? If people aren't familiar with PEG, it's polyethylene glycol, but what that really does is it extends the half-life of the drug, and it actually makes it a little less immunogenic in my world. So it's got a dual purpose. It helps increase the pharmacokinetic of the drug, and then it also should prevent maybe antibodies forming against it. Now, sometimes that's true. Certainly, it does extend the half-life, undoubtedly. It doesn't always make it less immunogenic. We're starting to realize that maybe you can actually generate an antibody against the PEG, even though PEG in and of itself is really an inert sort of compound that doesn't trigger immune responses. But I think that's a good, this is this is a really cool topic, John. I think I don't know how we want to wrap up, but I would like to stop here because the next wave is a full-on conversation. We're going to start talking about ADCs, combining them, cell and gene therapies, and further advancements. I don't think we have time to cover that today, John. But
I agree with you, and I think it's also worth talking about biomarkers and how they're a different
so we're going to put a pin in it. So it kind of brings us up right into about the 2000 to 2010 era in my mind. Even though ADCs start to pop up, they really don't get until 2010. Maybe I don't know how you want to wrap this up. I'll say that for me, somebody who's if I put myself in the 2010 range, I'm actually starting my CRO career. I'm doing things in the biologic space. We're doing ELE spots, but really the large molecules are still not quite. They're kind of coming to fruition, but it's still, even in 2010, it's 70% small molecules. And and that's even to take it a step further. Not only is it feeling like when I first joined the CRO space, I get told, well, why are you doing that? This is the beliefs back 20 years ago, right, or 15 years ago. Yeah. Is that because you can't make it in pharma? You're a bottom feeder. That was sort of some of what was said to me. But the reality was, I went because it was an excellent opportunity to leverage all of my immunological skills to help support like dendritic cell trials and things like that, which we'll talk more about how that set the stage for our current cell and gene therapies. But it's a wonderful topic, John, and I'm excited to pick it up on our next.
Yeah, and so I'll just close out with, if we say 2010, I think for me as an LCMS person, we're still in fairly conventional small molecule territory. Like say, you're starting to see other matrices come on board, mostly liquid for us, so we're not getting into the tissue piece, but we are starting on small molecule.
Are triple quads around? Like, is it a 4,000? What instrumentation?
So 2010 would probably. Oh God, it's mainly 4000s.
Yeah.
Then
That's a triple quad, right? That is a triple quad.
Yeah. So basically, so just to touch on this very quick, triple quads are really the dominant technology for quantitation, and have been, really from the 90s. But it's really been improvement in the optics of transmission through the triple three quadrupoles, but also the introduction of ions into the mass spectrometer in the first place. There's been a lot of focus in that interface. So ion spray, although it came on board in 1990 or thereabouts, really it's still the gold standard in terms of interface, but there's been gradual improvements to how that's been.
So the big leap doesn't come until, right? You're still not leaping for another few more years when you get into that 5,500, 6,500.
So 5,000s probably aren't too much more behind 2010. I'm not.
To me, it wasn't a big leap from 4 to 5, maybe, but it seemed like the leap was to the 6,500.
The big leap was the 365 to the 3,000, then the 3,000 to the 4,000. Okay. The 4,000 was huge because what it was, where the 3,000 had been spraying at the orifice, the 4,000, I'm going way down. The 4,000 was spraying parallel to the orifice, and that took out a lot of the junk. You're preventing junk going into the mass spectrometer. That was actually a huge piece of it. Then they've slowly looked at opening up the front end, so they've got increased pumping to get more of the ions of interest into the mass spectrometers. And so the 5,000, 6,500 and beyond are really more in the 2010 onwards.
Yeah,
but let's talk about that.
We're going to cut it up, but the 2010 flow cytometry, like the ECL instruments of today, are still kind of similar. They've advanced a little bit more. They might have taken a read time of 6 minutes. It's down to a minute, right? But they were still pretty good. Flow cytometries, that has really taken off. I think we were about 8 to 10 color. We're up to 25. So, John, this has been fantastic.
Sure. I'm going to just quickly say on the mass spec side, I think for next time, like say, the triple quads are really they just got more sensitive with time due to obviously improvement in engineering. I will touch on high res, as that's probably the big deal in terms of the last decade and where that started to contribute, because obviously we're going to touch on large molecule by LCMS, which we talked exclusively small molecule. Large molecule is a big part of this.
And Don Dufield would say, Don would tell you, why are you in 2010? I was doing that in 2000, right?
Yeah, no, understood, but I'm talking across an industry, so
yeah. And then likewise, I'm going to say spectral flow is up to that. That's our advancements. But John, let's
what a really great podcast. Can't wait to pick up part 2 of our evolution of drug design and its impact on bioanalysis. But John, this is a special time of the year. I love July 4th weekend. It's coming up. I got a couple extra days off. I'll let you start, but I got a jam-packed 4th of July.
So for us, well, obviously I'm British, so I don't really care about July 4th, but I do like the day off, so I'm not gonna complain about that. But our eldest son and his wife are coming over this weekend, which will be really nice. And so we'll have a nice family weekend. But that isn't my big news. For us, the big deal is later in July. So I've talked about the Chameleons on the podcast a number of times as a band who I've liked for years, and they were active in the 80s, split up, reformed around 2000, split up. But Mark Burgess, who is the lead singer of the band, has kept the name alive as Chameleons Vox for several years, and then he's since been joined by one of the guitarists, so they've readopted the Chameleons name. So they're actually looking to tour the US in the autumn. But for British bands coming to the US, that's a pretty expensive deal with visas and all sorts of, obviously, travel, even getting here. If we look at what airfares are like at the moment, that's going to have a huge impact on them. So Mark Burgess is actually putting together an acoustic tour where he's touring by himself to try and raise money for this autumn tour of the full band. So he put a post on Facebook saying, if anyone's interested in hosting a concert, let me know and we'll put a tour together. So I emailed him and said, yeah, come to Trumansburg, New York. We can host you. So July 22nd, Mark Burgess is first of all, he was saying, I'll play anyone's living room or wherever. And I thought about the house and garden and thought the logistics of doing something here, that's a nightmare. So we actually talked to the local brew pub, and he's playing the local brew pub on Friday, July 22nd, and then we're hosting him at our house. And so I think he plays a date in Boston on the 21st. It's Trumansburg, July 22nd, and then Buffalo, July 23rd. So yeah, we'll be hosting a musician from Manchester for a night, and hopefully have a good night of it, you know.
Well,
so
John, that sounds awesome. I mean, congrats, right? Like, this is a musician and a band from the 80s, right? That you probably grew up liking, and now you're John. Look at this. You're going to be hosting him, and this is like I don't want to say fantasy, but this must be a little bit of like a surreal to you, right? Like, that's got to be kind of cool.
Yeah, it's great. I mean, I've been sort of wandering around the area putting posters up.
And, you're like a 21-year-old, John. This is great.
Do you
got to staple in a bunch of flyers. You staple them on or how do you put them up?
No, just pin them up. To be honest, I think my eldest son is more excited than me. I've seen the Chameleons multiple times. I've seen Mark Burgess play multiple times. So it's like, although I'm doing it, it's like I haven't. It's just like
meet him, right? So if I understand you correctly, you're gonna like actually host him. So this is a whole. You've never really met him. You're gonna break bread with them and hang out.
I've met him at concerts and outside, and several years ago, my wife and I actually ended up backstage with the band in Rochester, and I got them to autograph stuff and things like that. So I've met him multiple times, but again, yes, it's always like as fan musician, so it might be slightly different after this. We'll just see how it goes. I've got no idea.
How do John? I don't even know how to follow this up, because I was like, I got a jam-packed 4th of July. All I'm going to do is light off a whole ton of fireworks, do a little water skiing. I'm going to Nebraska. One of my wife's families lives on a little lake out there, so we do some tubing and water skiing. I think I've gotten fit enough again. I think I can water ski rather than tube. But man, I don't have any rock stars or celebrities, but I will light off a lot of fireworks and eat a lot of good food.
I just wanted to let you know, because it should be a lot of fun. You know, because the thing about the Chameleons is that they never made it in terms of big money, but they've got this really strong cult following. So so obviously as this tour's been put together, there's a Facebook page where people post about it, but there's people crawling out the woodwork to go to this concert at a local brew pub. So it's
John, this is Jeremy. I don't often chime in, but this is awesome. That is awesome.
It's a shame that you're not close. I'm not close up to get people to come over, but sadly that's just the way it goes.
And John, some of my best rock or any sort of musical moments are in those small venues. Those tend to be
sure. Yeah. So another part of this is the brew pub is called Garrett's Brewing Company now, but it was actually used to be called a venue called the Rongovian Embassy to the United States, and it was like I think it is set up in the 1970s. This fairly hippie hangout, and it had been going for years under that name. It is known across the country as the Rongo. People, if you mention Trumansburg, they I've been to the Rongo. It's well known for that. So it's really nice, and so a lot of bands used to play there. Again, it's mostly local people, but it's really nice to bring some from the UK and have them play the Rongo.
Well,
and then the other part of Trumansburg, is I don't know if I've mentioned this before, but the Moog Synthesizer, the Moog Synthesizer that a lot of musicians use.
Yeah, I think so.
So Bob Moog's original workshop was in the centre of town. It's actually now a restaurant that we go and eat at every Friday night. So there's a bit of a musical legacy in this village,
Yeah, it's a rich area, for sure. So
yeah,
John.
Anyway, that's our big July thing.
Not that it's a competition, but you taught me. I was going to tell you, I got some great. My buddy of mine, he knows somebody who owns a fireworks stand. So the last few years, I've been getting a wonderful, a solid 2-hour show of fireworks that is impressive for my son, my wife, and my in-laws.
And to be honest, To be honest, I would love that.
It's
I've
great.
always loved fireworks. It's like if I could be there, I would.
No, I mean it's to the point where all of my in-laws' neighbors kind of there's like at least 15 or 20 of them who are expecting us to go and light the sky up for a few hours.
Yeah,
and we won't disappoint. John, I want to touch on some food I've been cooking. So my son,
can I just touch on the firework thing very quickly? Because several years, one of the local wineries to us has done a barbecue and fireworks. That's always a really nice event. Sadly, COVID has made that really difficult for them to do, and it's not happening this year. So hopefully next year they'll be able to pick that back up. But it was like, it's they have the winery and they have the little restaurant, and they had like a pond, and they have like fireworks happening over the pond. It's a really nice setting, and it's such a shame that they're not able to do it.
My wife's hometown puts on a big fireworks show, so that's July 3rd they do it. So I'll go to a nice it's a wonderful show. It's huge. And then I'll light off for a few hours on the 4th. But John, my son took a job at a place called Minsky's Pizza. I don't know. So I have not been using my Uni grill, and I have not been making pizza because he comes home a couple times a week with this one. We love Minsky's Pizza, so I've been like, I'm jonesing for my Uni, but I just can't do it when I have all this pizza coming in. I got to watch my waistline. So I've been using my precision cooker a lot lately, and man, do I love that thing, John. I've been making fish and steaks. I mean, it is so unbelievably tender when you sous vide a steak for a couple hours and then you sear it either on a grill or my cast iron. Yeah. I mean, even my wife and son are like, wow. I've earned now my steak badge from my son, so I've got a whole host of smoking badges. He's officially said I've earned a ribeye badge from him now.
That sounds impressive. Yeah, at some point I'm going to need to check that out.
Oh, John, you're going to be fascinated if you get a precision cooker. It is something that for a while I didn't use it. My wife was like, now she's like, you didn't like my birthday gift, one of those stories, and now I use it routinely. Yeah. So, John, happy 4th of July to you.
Thank you.
Great show, and we'll pick it up soon. Good luck with the concert. Congrats on that. Excited for you.
Yeah, thanks a lot. We'll talk between now and then, obviously. But yeah, thanks. I think I had a great show today, and look forward to picking this topic up next time.